DNA-dependent SUMO modification of PARP-1.
Written by Zelio et al.
Presented by Farhan Mirza,
Adam Taoudi,
and Zarrin Kadir.
PARP-1, poly-ADP ribose polymerase 1, also known as NAD plus ADP ribosyltransferase 1, or poly-ADP ribose synthase 1, is an enzyme that works by modifying nuclear proteins by poly-ADP ribosylation. SUMO proteins, which are small ubiquitin-like modifier proteins, work by covalently attaching and detaching from other proteins. SUMOylation is a post-translational modification involved in transcriptional regulation among others, such as nuclear cytosolic transport, transcriptional regulation, apoptosis, protein stability, response to stress, and progression through the cell cycle. PARP-1 is a transcriptional coactivator of hypoxia-responsive genes. Modification of PARP-1 by attachment of SUMO leads to a change in its function and promotes induction of heat shock. The report is suggesting the SUMOylation of PARP-1 is influenced by DNA. It also shows how SUMOylation in vitro is enhanced by binding to intact DNA, so PARP-1's catalytic activity isn't stimulated, which is only activated when damaged DNA is present. PARP-1 has many ways to identify DNA in its different forms. This allows it to single out different aspects of its own function.
SUMOylation reactions were carried out in a solution containing many components, including 220, a buffer, and 240 nanograms of DNA. Afterwards, analytical reactions were carried out in a total volume of 20 microliters, 3 microliters of which were loaded onto either 10% weight per volume PARP-1, F1 and F2 polyacrylamide gel, or 6% weight per volume full-length PARP-1 polyacrylamide gel. For mass spectroscopy, the reaction was scaled up to 45 mL for PARP-1, F1 and F2, but scaled up to 1.3 milliliters for the full-length PARP-1. For nuclear magnetic resonance analysis, the reaction was scaled up to 1.4 liters. DNA binding assays were performed with 60 picomoles of 15 base pairs duplex DNA. Unlabeled DNA acted as a negative control. The DNA was then incubated with the protein in a binding buffer on ice. This mixture was then added to 10 microliters of streptavidin agarose that has been blocked with a blocking buffer known as bovine serum albumin into a binding buffer and incubated with rotation at 4 degrees Celsius for an hour. The beads were washed 3 times with binding buffer, and any bound material was extracted by boiling it in an STS loading buffer. Then the proteins were incubated with 10 microliters of SUMO2 agarose, repeating the previous step. Interactions between the proteins were assayed in the same way with different concentrations. The protein mixtures were incubated on ice for an hour and added to the beads, and then they were washed and bound as before. The bound material was analysed by Western blotting with the relevant antibodies.
SUMOylation in vitro is enhanced by binding to intact but not to damaged DNA. This is different from the mechanism by which DNA damage stimulates PARP-1's catalytic activity. Now, SUMOylation does not interfere with the catalytic or DNA binding properties of PARP-1. As you can see in the diagram given in this presentation, in part A you can see that SUMOylation of F1 and F2 is inhibited by single-stranded breaks. You can actually see the structure of the nicked dumbbell. In part B of the diagram you can see that the addition of the nicked dumbbell inhibits the stimulatory effect of a supercoiled plasmid DNA on the SUMOylation of F1 and F2. In part C you can see that the F2 domain is sufficient for the stimulatory and inhibitory effects of DNA on SUMOylation. And in the last part D you can see that inactivation of F2 abolishes the effect of DNA on SUMOylation.
From the findings, it can be delineated that different parts of the protein PARP-1 are used to detect the state of DNA. For example, if it is intact or damaged, or single or double-stranded. PARP-1 was stimulated by DNA without any usual features, which, because the protein's F2 domain can differentiate between damaged and intact DNA, the research can support the model where the F2 domain is what interacts with undamaged DNA. In this instance, the protein was efficiently simulated. The function of the F2 domain tells us that it forms robust contacts between the protein and the uninterrupted sugar phosphate backbone of DNA. The fact that F2 was overcome by long stretches of double-stranded DNA suggests that additional domains also contribute to its recognition, with the WGR domain as the most likely candidate. The way DNA structure affects PARP1 implies there is a level of signaling across the protein.